Bismillah ar-Rahman ar-Rahim. Welcome again to this important new video. Our subject today is about an important pathologic process that happen in many diseases. This important pathologic process is called thromboinflammation. So our subject today is about thromboinflammation.
To start with, we have to differentiate between two concepts, hemostasis and thrombosis. Hemostasis means the process of clot formation under normal physiologic situations, aiming to stop bleeding, like in blood vessel damage, as in wounds. While thrombosis means clot formation under pathological situations, like in atherosclerosis, plaque rupture, deep vein thrombosis, pulmonary embolism, stroke, and other examples. And I think all of us remember studying in the first years in medical school about Virchow's triad, which is responsible for thrombosis in pathological conditions. This triad shows three important factors that are responsible for this process, which are stasis of the blood flow, endothelial injury, and hypercoagulability. And as our subject today is about thromboinflammation, we can say that inflammation at least affects two factors, which are endothelial injury and hypercoagulability. An example of the process of thromboinflammation is the association between thrombosis with autoimmune diseases, and actually there were many researches that tried to study the relation between autoimmune diseases and venous thromboembolism. We can see here this paper in American Journal of Cardiovascular Disease 2021, Autoimmune Diseases and Venous Thromboembolism, review of the literature. Another paper was done in 2021 in Circulation, which says risk of venous thromboembolism in autoimmune connective tissue diseases, a population-based retrospective study. And this paper concluded that various autoimmune connective tissue diseases were significantly associated with the risk of venous thromboembolism. So we can expect easily that inflammation will activate endothelial lining and exposing proteins Willebrand and collagen, which will attract platelets and tissue factor, and then this will activate factor 7 and coagulation. Platelets will form a thrombus when exposed fibrin, and this thrombosis, on the other hand, will then activate innate immune system by the process known as immunothrombosis and netosis, which will in turn will activate thrombosis, and so we will get vicious circle between thrombosis and inflammation. This paper, which is published in Blood in 2019, which says that thromboinflammation challenges of therapeutically targeting coagulation and other host defense mechanisms. In this paper, they said that thromboinflammation occurs in a broad range of human disorders. These disorders is well recognized clinically in the context of superficial thrombophlebitis. We have inflammation and thrombosis. However, this thromboinflammation is more dangerous when it involves the microvasculature of injured tissues and organs. And examples of this thromboinflammation of the microvasculatures we can see in the cases of sepsis and ischemia, reperfusion injury, and also in organ transplant rejections, major trauma, severe burns, antiphospholipid syndromes, preeclampsia, and sickle cell diseases. In all these situations, we have also loss of normal antithrombotic and anti-inflammatory functions as a central part of thromboinflammation. These all will lead to dysregulation of coagulation, complement, platelet activation, and leukocyte recruitment in the microvasculature. While understanding the process of thromboinflammation, two concepts we have to remember always. The first one is that thrombosis is basically a way of defense against microbes, and the second important concept is that managing inflammation in some cases, like in Behcet disease, has the priority over antithrombosis in managing thrombosis. Researchers have found that the active interplay between thrombosis and inflammation, thromboinflammation, is a major underlying pathway driving cardiovascular diseases, autoimmune diseases, and most recently also COVID-19. This nice paper that was published in July 2022 in arteriosclerosis, thrombosis, and vascular biology. Its title is Thromboinflammation from Atherosclerosis to COVID-19. I found this paper very informative and nice to discuss. This diagram from the mentioned paper, where we can see the active interplayers in the process of thromboinflammation. We find here the neutrophils, the monocytes, endothelial cells, and platelets, where NETosis or extracellular neutrophilic traps form a platform with von Willebrand factor, where platelet form a string above it, and attached to it is the tissue factor, which will stimulate coagulation. Neutrophils, which are the most abundant white blood cells in humans, take a central role in the process of thromboinflammation. This is because their ability to trigger or form an organized cell death pathway with the release of decondensed chromatin structures, which is called neutrophil extracellular traps. This chromatin structure is decorated with histones and cytoplasmic and granular proteins. These neutrophil extracellular traps exert cytotoxic, immunogenic, and prothrombotic effects, accelerating disease progression, and play a central role in thromboinflammation. The process of NETosis, or the formation of neutrophil extracellular traps, as I explained already in other videos, include many steps, and one important player in these steps is BAT4, or protein arginase deaminase 4, which catalyze the citrullination of histones that lead to chromatin decondensation. Also, BAT4 plays an important role also in inflammasome activation in neutrophils. Again, as I explained before in NETosis videos, BAT4 drives chromatin decondensation by catalyzing citrullination of positively charged arginine residues abundant in histone, changes them to citrulline, an amino acid that lacks a charge. Loss of the positive charge will loosen the histone interaction within the nucleosomes and with DNA, and will lead to histone proteolysis and unwinding of chromatin into nets. Again, in the same diagram, we can see the role of BAD4 in the process of netosis and in activation also of inflammasomes, and later on we'll know its role also in preventing cleavage of von Willebrand factor. Actually, BAD4 play an important role in linking the immunologic function of neutrophils with procoagulant and proinflammatory activities of monocytes and the platelets, and this role will foil thromboinflammation. And as I explained before, NETs or neutrophil extracellular traps provide a large platform that is decorated with histones and cytoplasmic and granular proteins. This large platform is harmful to surrounding tissue and also present new antigens that will later on augment the process of thromboinflammation and will induce autoimmune diseases. Within the blood vessels, NETs will preserve a function like von Willebrand factor because both together form a big platform for platelet adhesion and coagulation initiation. And as I said before, the functions or the role of BAD4 include its inhibition to von Willebrand factor cleavage, allowing the platelet strength to persist near the vessel wall in the presence of BAD4. Again, in the same diagram, we can see the important role of BAD4 in netosis, in inflammasome, in preventing cleavage of von Willebrand factor, so that we have a big platform formed from NETs, from neutrophil extracellular traps, in addition to von Willebrand factor, where the platelets can form a string and attach it to the tissue factor that will stimulate coagulation. Tissue factor containing microparticles that are deposited in NETs or neutrophil extracellular traps come from multiple sources, where monocytes are the primary source of these procoagulant microparticles. However, there are other sources like malignant cells. Actually, netosis and NET-associated tissue factor was found to be increased with increased level of systemic inflammation and interleukin-1 beta, indicating a common regulating pathway. We know that interleukin-1 beta is a product of activation of inflammasomes through active caspases. That's why activation of inflammasomes triggers the release of tissue factor from activated macrophages and monocytes. So we can see here the main source of tissue factor is from monocytes, and also activation of inflammasomes will result in activation of interleukin-1 beta, which is correlated in many researches with the level of tissue factor. So activation of inflammasome will help in providing tissue factor. As regards the role of platelets, platelets are anucleate cells produced by megakaryocytes, and they play an essential and important role in infectious diseases. They represent a part of the effector cells, which play a critical role in bridging hemostatic, inflammatory, and immune processes. Platelets constantly actually scanning in the whole blood for areas of injury or inflammation as they circulate in the vasculature. So we can understand that platelets are cellular mediator not only of thrombosis, but also platelets are immune cells that initiate and accelerate the inflammatory conditions in the vascular system. Actually, platelets are linked to the pathogenesis of inflammatory diseases such as atherosclerosis and in infections like malaria and in transplant rejection and in autoimmune disease like rheumatoid arthritis. So platelets in all these situations play an immunological functions which are protective, but sometimes platelets contribute to adverse inflammatory outcomes. So in certain conditions, platelet activation may be injurious to the host, which will result in exacerbation of inflammation and promotion of endothelial damage and thrombosis. P-selectin is a cell adhesion molecule that play an important role in the process of thromboinflammation. P-selectin being stored and released by platelets and Weber pallet bodies of endothelial cells play an important role in leukocyte recruitment and activation. P-selectin interact with its ligand P-selectin glycoprotein ligand 1 and stimulate P-selectin shedding. Actually, soluble P-selectin retains many of its procoagulant and stimulatory properties, and elevated soluble P-selectin in the plasma are associated with increased risk of myocardial infarction, stroke, cardiovascular diseases in humans. In monocytes, binding of P-selectin triggers rapid surface exposure of tissue factor, which will then activate factor 7 and coagulation. So again, in this diagram, we can see that platelet activation, as a result of finding something wrong while they are circulating in the blood, when platelet become activated, they express P-selectin from its granules inside, and this P-selectin cell adhesion molecule will bind to its ligand, P-selectin glycoprotein ligand 1, on the surface cell membrane of neutrophils and monocytes, and this will result in reprogramming of these cells. So in neutrophils, activation of neutrophilic cells through platelets will result in activation of the process of netosis, or formation of extracellular neutrophil extracellular traps, and this will also preceded by activation of inflammasome. So as I said before, PAD4 is important for the process of netosis or formation of neutrophil extracellular traps, as well as it's important for inflammasome activation, and also thrombin generation will result localized to the surface of activated platelets. This thrombin generation will result from tissue factor released from monocytes through activation by platelets, and this thrombin will activate the endothelial cells, which will result in rapid secretion of von Willebrand factor from Weber pallet body, and once released, von Willebrand factor will stand into long strands and exert multiple prothrombotic effects, forming a platform attaching nets to the vessel wall. Also, the inflammasome, through generation of active caspase-1, will result in the release of the important vascular cytokine interleukin-1 beta, which will lead to expression of adhesion molecules E-selectin and ICAM1 on endothelial cells, which will result to more leukocyte recruitment. And actually, the interplay between platelets and the immune cells can, partially through interleukin-1 beta production, present an autocrine feedback loop which can snowball into a cytokine storm. As I said before, PAD4 has a third function in addition to process of netosis and inflammasome activation. PAD4 also inhibits what's called ADAM TS13. This ADAM TS13, which is abbreviation to disintegrin-like metallopeptidases with thrombospondin motif type 1 member 13. This is responsible for cleavage or cutting of the Willebrand factor. So by inhibiting this enzyme, we will have the longest strands of Willebrand factor and will allow platelet strengths to be present and persist throughout the vessel wall. So the von Willebrand factor will form as a big platform for attachment of the platelets and for connecting the nets to the endothelial lining. Also, histones as well as cytoplasmic and granular proteins entangled in these neutrophilic extracellular traps or nets have many effects following the pathological response. Also, platelet activation and binding to monocytes will result in monocyte activation and inflammasome activation in monocytes. Activated platelets also will stimulate tissue factor synthesis inside the monocytes and its release in vesicles outside. This tissue factor will stimulate thrombin generation following a systemic procoagulant state and further platelet activation. So we will get in like a vicious cycle of thromboinflammation. Again, over time, an increased tissue factor expression from the monocytes and its shedding into vesicles in the extracellular space from the monocyte surface will become the primary source of blood-borne tissue factor, and this tissue factor will activate factor 7, which will later on form thrombin, and so we will get at the end the generation of large amount of thrombin. Activated platelets release many activating agents, in which serotonin is one of them. Serotonin not only induces vasoconstriction, as we all know, but also serotonin amplifies platelet and endothelial activation. Actually, serotonin facilitates Weber palate body exocytosis. As a more explanation, inflammasomes already are a family of macromolecular ring-like multi-component protein structures that are formed as a result of activation of innate immune cells. They were being studied in monocyte and macrophages. However, in recent researches also there was evidence for inflammasome assembly in neutrophils. So if we put the cellular components aside, the process of thromboinflammation is orchestrated by a complex interplay between coagulation, cascade, complement system, and cytokines. And the primary cytokine here is interleukin-1 family. Many of these regulatory proteins are synthesized from inactive precursors, cytokines, and require proteolytic processing to attain a biological function. Caspase 1 is a major intracellular protease implicated in processing of pro-interleukin 1 beta to active interleukin 1 beta, and this caspase 1 already is a workhorse in inflammasome. So once activated and released, the interleukin 1 beta is the major pro-inflammatory cytokines that will drive the synthesis of endothelial adhesion molecules, which will recruit leukocytes. Endothelial adhesion molecules like E-selectin, ICAM1, ICAM intracellular adhesion molecule 1, and VCAM1, vascular cell adhesion molecule 1. The multimeric glycoprotein von Willebrand factor recruits and activates platelets through binding to platelet glycoprotein 1b alpha. Von Willebrand factor can be found in the same location as P-selectin, and also it supports platelet and leukocyte attachment in a similar way. If left uncleaved, von Willebrand factor will form long strings that will be attached transiently to the endothelial surface. The ultra-large von Willebrand factor will break the red blood cells, producing schistocytes, as situation in thrombotic thrombocytopenic purpura, where there is decreased activity of ADAM TS13 responsible for cleavage of von Willebrand factor. So in our case of thromboinflammation here, the fractures in red blood cells will result in heme leaks, and these heme leaks will activate the process of netosis that will amplify more the thromboinflammatory process. And as I described before, this large von Willebrand factor will be stretched in longest strengths and will lead to initiation of platelet strengths above it and microthrombosis. Histological analysis of the thrombi found that it's formed from a localized collection of fibrin, nets, and von Willebrand factor in the solid matrix of thrombi. Von Willebrand factor exerts multiple thrombus stabilizing effects by forming a bridge between the vessel wall and the nets, while histones released with nets promotes the von Willebrand factor release from endothelial cells and platelet activation. Actually, there is a direct interaction between von Willebrand factor and the nets. Both DNA and histones bind von Willebrand factor, thus retaining nets at the site of inflammation. As an example of the process of thromboinflammation in inflammatory disease, we have Behcet disease, Behcet syndrome as an example. I get this paper published in Frontiers Immunology 2019, Behcet syndrome as a model of thromboinflammation, the role of neutrophils. Another paper published in Journal of Korean Medicine Science 2020 says neutrophil-mediated thrombosis and netosis in Behcet disease hypothesis. So in these papers, they are telling that, as we all know, Behcet disease is a complex autoinflammatory disease, and thrombosis is a significant aspect of the disease. Actually, neutrophilic venous thrombosis is a probable primary mechanism in Behcet disease, whereas hyperactive neutrophils in the vessel produce reactive oxygen species and nets and augment inflammation, cause endothelial damage, and also triggering thrombosis cascade. The platelets are recruited to the site of damage, degranulate and aggregate, which further induce inflammation and thrombosis. Also, the produced thrombus gets more resistant to plasmin-induced lysis through fibrinogen carbonylation with reactive oxygen species produced by hyperactive neutrophils. So, as a conclusion, neither thrombosis nor inflammation occur in isolation. They accompany each other. So, clot formation is no longer understood to contain only cleaved fibrinogen and with plasma proteins. And clot DNA content now has to be considered for clot lysis. Also, platelet plugs are far from being only platelets cross-linked by fibrinogen. And as interesting finding that mice lacking both fibrinogen and von Willebrand factor were still able to produce thrombi after injury. Also, in deep vein thrombosis, it requires not only platelet and red blood cells, but also monocyte and neutrophils. So the classical cellular layers of inflammation fully intermingle with those of thrombosis, justifying the term thromboinflammation.
Lastly, I hope that you understood the process of thromboinflammation. And again, I want to remind you to subscribe to the channel and activating the alarming bell if you are interested in getting any new videos. And thank you.